My dear students, it's Snacks Biology and I'm Hemant Naik, your biology teacher. One thought for the day that if you're facing some problems, you feel that I am frustrated, I can't do this, I can't do that. It is not like that. When the problem will come, it is make you more mature, more confident, more sincere. It will develop your inner quality by which you are able to face the challenges in your future life. Okay, it's today's thought. Now be prepared when the difficulties will come. Bless to the God, give the difficulties, make me more mature, more sincere, and more energetic to face the difficulties. Now, today we are going to start about the new concept and important concept, that is Operon concept. This Operon concept is very, very important for the CBSE board exam. All of you know. That's why make it very clear. See the full video. If you are very clear, then don't forget to subscribe my channel. Then love, what is the operon? Before starting the operon concept, we will start what is the operon. Operon means it is a set of the genes. You gene, it is a what is the gene? Gene means segment of DNA which is carry the characters. That is called the gene. And here what? Operon means set of the genes. But in our book, CBSE book, what is in our syllabus? It is lac operon. Lac operon means what? The lac operon is a set of genes that are specific for the metabolism of lactose. Lactose what? Lactose is nothing but a disaccharides. When it will break down, it will form the two monosaccharides. You know that monosaccharide and disaccharides. This monosaccharide is glucose and galactose. Then this where this lactose is there? Suppose you think there is a two media. In one culture, the bacteria with the lactose and one another culture bacteria without lactose. When it is the difference of the culture you are seeing, no, दो कल्चर में एक में लैक्टोज है और एक में लैक्टोज नहीं है. Where the lactose is there, what will happen generally? The bacteria will secrete the enzymes which will break down the lactose. And जिसमें lactose नहीं है तो कोई सवाल ही नहीं पैदा होता, क्योंकि it will not synthesize the enzymes which will break the lactose. Now start the terms which you should familiar with that. First, like that one diagram is there in your NCERT textbook. What is this? It is a DNA. And what is the segments? It is the segment are called the genes. But don't forget that all the parts of the segment of DNA not responsible for the production of the enzymes or production of the protein. Some segment only responsible for P, or it is called, what is that called? You see, promoter gene. I for regulator or inhibitor, both the functions it will do. O for operator, and ZYA, these are called the structural genes which is responsible for formation of the enzymes, that is proteins. So first, from what is lac operon consists of, what I told you. See, one is one regulator gene or inhibitor gene, that is I, one promoter gene, that is P, one operator gene, that is O, and three structural genes, that is Z, Y, A. These are the three structural genes. First of all, what is the regulator gene? That is I. The gene codes for the protein known as the repressor protein. First, DNA, it is a I is a segment of DNA. It will forms the, definitely it forms a mRNA, that is called repressor mRNA. After that, it will forms a repressor protein. That means this inhibitor is a segment of DNA which is produce a protein that is called repressor protein. Clear? After it is only the segment. This is a, don't forget, it is a segment. It is a DNA and each part one segment, and they are responsible for that particular work. After that, promoter gene. P means promoter gene. What is this gene? Is possess the site for the RNA polymerase. You know, when a DNA will produce the RNA, it will take the help of a enzyme that is called RNA polymerase. When DNA, don't be confused, DNA produce the DNA, then DNA polymerase. Here what? This DNA segment produce a mRNA. That's why RNA polymerase. This site will controlled by the promoter. That means promoter gene, the gene possesses the site for RNA polymerase attachment. Because without the attachment of RNA polymerase, a DNA cannot produce the RNA. After that, operator gene, O, you see here operator. What it will do? It will interact with the protein molecules. What protein molecules? What is produced by the? What is produced by the? What is this gene? It is I, regulator gene. Regulator gene, what protein will form? This protein is a regulator protein which will prevent the transcription. Transcription means what? When a DNA will produce the RNA, it is called transcription. When the RNA will produce the protein, that is called the translation. Clear? Where the when it will produce the enzyme, when they will transcription occur to form the mRNA, and mRNA will follow the translation, then only protein will form. This is controlled by the operator gene. And structural gene, you know that is a ZYA, three structural genes, and they will produce the different types of proteins. What to understand in this? We understand that operon is nothing but a segment of DNA genes present in the DNA. Lac operon means because it is related with a saccharide that is called lactose. This lactose, what will happen? It will break down into the glucose and galactose. When it is present, the system will work. When in a media, when the lactose is present, the lac operon will work. When the lactose is absent, then lac operon will not work. Not work, it means these three are become not produce any enzyme. Now we will see details of the lac operon. Are you clear this concept? Okay. Now what you will try to understand? We try to understand which is switched off and which is switched on. यहाँ क्या लिखा है? Where it is, what is written? Switched off. It is switched on. What does it mean? When it will occur? First, what is switched off? In simple meaning, what you understand? When the enzymes are not produced to break down the lactose. That means जब enzyme produce होगा ही नहीं, तो इसका मतलब क्या है? Lactose medium में नहीं होगा. तो in absence of lactose, it is switched off, because when कोई भी मीडियम में लैक्टोज है ही नहीं, तो बैक्टीरिया का कोई मतलब नहीं बनता कि उसको एंजाइम प्रोड्यूस करे, तो वो क्या होता है? वो एंजाइम अपना प्रोड्यूस नहीं करेगा. तो दैट इज कॉल्ड द स्विच्ड ऑफ. और वी कैन आल्सो कॉल द नेगेटिव कंट्रोल रेगुलेशन. नेगेटिव कंट्रोल और नेगेटिव रेगुलेशन वी कैन से. तो हाउ इट इज हैपेंड? ऐसा क्यों हुआ कि इंड्यूसर, दैट मीन्स लैक्टोज नहीं है, तो अपना जो एंजाइम प्रोड्यूस होने वाला था वो नहीं हुआ. देखो, वो P है, I है, I is a, you know, regulator gene. Regulator gene is produced mRNA, because it is a DNA, it will produce mRNA. That is called the repressor mRNA. After the repressor mRNA, it will produce a protein that is called repressor. Don't forget, it is a DNA. It produces, it is a fragment of DNA or segment of DNA. It produces a repressor mRNA and it produces a repressor. That means a protein. This protein, what we know that what is the operator? This operator will bind by the repressor. क्या bind करेगा? It will bind the RNA polymerase. When it will bind the RNA polymerase, this ZYA will not produce any enzyme. In simple, what you will tell, the repressor binds to the operator region and it prevents the RNA polymerase from the transcribing और from the transcription. वो जो transcription होने वाला था, वो transcription नहीं होगा. Why? Because the repression जो एक प्रोटीन डेवलप हुआ है, वो प्रोटीन क्या है? ऑपरेटर को बंद कर दिया. बंद करने के बाद ये क्या हुआ? जैसे बंद कर दिया, तो उसमें ये जो कंट्रोल कर रहा था, किसको कंट्रोल कर रहा था? ये कंट्रोल कर रहा था कि आरएनए पॉलीमरेज को. और वो आरएनए पॉलीमरेज वहां सीक्रेट नहीं होगा, तो वो प्रोटीन सीक्रेट नहीं होगा. ये ऑफ कंडीशन है. हम स्विच्ड ऑन कंडीशन है. स्विच्ड ऑन का मतलब है इंड्यूसर है. इंड्यूसर मतलब क्या है? यहाँ लैक्टोज है, मीडियम में क्या है? लैक्टोज है. तो लैक्टोज में जब है, देखो PIPOZY, ये उसका सीक्वेंस है और एक डीएनए है. इसमें जब वो इंड्यूसर है, तो आई जो है वो एक यहाँ इनहिबिटर, मींस कोई भी काम को बंद करना है तो इनहिबिटर है. रेगुलेटर का काम कर रहा है, तो रेगुलेटर जीन है. वो क्या प्रोड्यूस किया है? एमआरएनए, रिप्रेसर एमआरएनए. रिप्रेसर एमआरएनए क्या प्रोड्यूस किया? रिप्रेसर प्रोटीन. ये जो प्रोटीन जो फॉर्मेशन हुआ, रिप्रेसर प्रोटीन, इसको इंड्यूसर जो मीडियम में था, वो इंड्यूसर क्या है? लैक्टोज जो है वो बंद कर दिया. इट विल स्टॉप द इंड्यूसर. डोंट फॉरगेट, व्हेन द इंड्यूस रिप्रेसर बंद हो गया, बाइंड हो गया, सीज हो गया, तो ऑपरेटर अपना फ्री हो गया, उसको कोई रोका नहीं अभी. तो वो operator will start working. When the operator will start working with the help of RNA polymerase, then it produces the 3 RNAs, and these 3 RNAs produce the first beta galactosidase, permease and transacetylase. Beta galactoside will break down lactose को break down करना है क्या? कि कोई भी चीज डाइसेक्राइड ब्रेक डाउन होने के बाद ही एब्जॉर्ब होगा, तो ग्लूकोज एंड गैलेक्टोज होगा. और परमेज, इट विल हेल्प टू द परमेबिलिटी. और ट्रांसएसिटाइलेज एसिटाइल ग्रुप को ऐड करेगा, जो हमारा बीटा गैलेक्टोसाइड हेल्प इन द डाइजेशन. वो अपना काम करने के लिए जो ट्रांसलेटरी हेल्प्स टू द बीटा गैलेक्टोसाइडेज टू ब्रेक डाउन द लैक्टोज मॉलिक्यूल्स। तो इसको एक सिंपल इक्वेशन में वी कैन ट्राई टू अंडरस्टैंड। रिप्रेसर प्रोटीन मींस दिस प्रोटीन कंबाइंस विद द ऑपरेटर जीन, यहाँ जीन लिख देता हूं, ये एक ऑपरेटर जीन, इट विल स्विच्ड ऑफ। स्विच्ड ऑफ का कंडीशन क्या है कि रिप्रेशन प्रोटीन के साथ जब ऑपरेटर जीन मिलेगा तो स्विच ऑफ हो जाएगा, तो तीनों में कोई एंजाइम सीक्रेट नहीं होगा। सेकंड कंडीशन क्या है, इंड्यूसर। कौन सा इंड्यूसर है भाई, इंड्यूसर हमारा लैक्टोज है। उसके बाद जो रिप्रेसर है वो कहाँ है, रिप्रेसर प्रोटीन को बांध दिया तो वो स्विच ऑन हो गया। इसका मतलब क्या है, जो तीनों एंजाइम है अपना अपना काम करना शुरू कर दिए। व्हाट यू अंडरस्टैंड द टुडेस लेसन, we understand that Operon is a system to metabolize the lactose. When in a medium, the lactose have the bacteria and lactose. Two things are there. Bacteria is organism which will use the lactose. How it will use the lactose? It is a system by the Operon. When this operon, when the enzymes become active, that means when it will active, when the lactose is present, then we call it a switched on condition. When the lactose is absent, which is called a switched off condition. And in the switched off condition, the enzymes will not secrete because operator will bind by the repressor protein. When switched on, when the repressor protein will develop, it is bind by a inducer or lactose. Then the three enzymes become continuously formed. Z, Y, A. Z will form the beta galactosidase, Y is form the permease, and A is form the transacetylase. This is the three enzymes which is formed and helps to metabolize the lactose. It is all about to the today's lesson, and it is a very, don't forget, it is a very important question in any aspect, in any form the questions will come. That's why these two equation what I told now, that is very important. What is that what I told? Once again I want to repeat, repressor protein, when it is combines with the operator, it is switched off condition. That means in this case the lactose is not present in the medium, and inducer combines with the repressor switched on condition, because here the lactose is present here. This is the two difference, on and off. And I hope you understand the concept very clearly. Thank you for watching.